Alex is co-organizer of this workshop, and she's an assistant professor in Earth System Science at Stanford University, and yet again, another engineer. Alex is interested in ecohydrology and remote sensing, ecosystems in the carbon cycle, the Earth, the universe, and everything. And so it's her time to speak.
Okay, so officially I guess it's about spaceborne observations, but I have the benefit of having lots of speakers before me talk up and increase the pressure on me, as this is going to be the quote-unquote remote sensing talk. So I'll actually focus mostly on measurements in the microwave frequency range for the primary reason that those are directly sensitive to water content itself. That's why the title was Observations of Water Content. But as Ashley pointed to a little bit at the end of her talk, and the other speakers as well, there's lots and lots of different remote sensing technologies that are relevant to sort of questions of ecosystem water responses. So I'm going to start this talk after a little bit of an introduction, just by hope these slides are not out of date, but we'll figure that out. By giving a little highlight reel of lots of different remote sensing technologies. So thankfully, we have experts in the room on every single one, but that's just to get you excited. And then I'll explain a little bit about why microwaves are actually sensitive and how microwaves are actually sensitive to water content, and ultimately then talk about where we go in terms of whether or not we can actually get these variables like leaf and xylem water potential that have been all over the so we'll get to the relevant variables. And then lastly, I'll talk a little bit about biomass. So let's just dive right in. And I think most of you are at least open to the idea of remote sensing, but I really like this figure for motivating the issues here. So here in the orange is the distribution across latitudes, across Earth, of most of our ground forest inventories. And those are mostly in the mid-latitudes, in places where there's a lot of money for science, basically, and lots of scientists to study things and go take measurements. Unfortunately, when we think about where most of the vegetation carbon on Earth is, it's in this blue line. It's in places like the tropics and places like the boreal regions. So we really have a bias in our in situ measurements, and we need measurements that can really span and cross all of the Earth and go to these regions, like the tropics and the boreal regions. And that's not just something about inventories, right? It's across sort of ecosystem variables. Just for the sake of completeness, Jan Laan very nicely made a similar plot, similar distribution plot of all of the P50 measurements in the TRI database, right? And you get the exact same distribution. So we have this biased set of estimates, and we need all of those estimates in the first place because there is so much variability in the response of different trees to water stress. Thankfully, every single one of the previous speakers has really highlighted these points, right? But this is a plot of just a coefficient of variation of P50 that Bill Anderegg previously put together across different groups. And you see even within species, there's significant variability. Within a PFT, you have as much as 50% relative variability in P50 values. So we really need measurements everywhere in order to be able to figure out how to scale across all of this variability on Earth. And that's where the remote sensing comes in. So I mentioned I'll mostly talk about microwaves today. They're directly sensitive to water content. That's the main reason. Another key advantage to microwave regions, microwave frequencies, is that they are relatively well able to penetrate through the atmosphere, and especially through cloudy conditions, unlike most other remote sensing types. So when you have fast-moving things like water in trees or in the soil, that's really important. And I just want to highlight relatively early on, right, this is a frequency range within the electromagnetic spectrum, as you see by this range of wavelengths here. But there's multiple microwave frequencies, there's multiple what we call bands. Unfortunately, I don't really have time to talk right now about all of these different bands and the differences. But for now, I just want to make the point that, yes, there's different frequencies. They different sensors at these different frequencies, and they all penetrate the canopy to sort of different extents. So for those of you sticking around the rest of the week, I'm sure that'll come up, but I just wanted to raise the point earlier. Okay, so this is where microwaves are in the electromagnetic frequency, the relatively long wavelengths. But I promise you, a quick highlights tour. And for that, we'll mostly focus on sort of visible and near infrared or infrared measurements that can do a lot of other things for us depending on the sensor types. There's a few sort of commonalities, one of which is, well, actually, before I go into that, one main commonality is that unlike the microwaves, you do have the disadvantage of being sensitive to cloud cover. But on the flip side, because of these shorter wavelengths, it's a lot easier to get higher resolution measurements. So that is a key advantage here, and you'll see that in these examples. So I'll talk a little bit about three quick things. We'll talk about evapotranspiration measurements. We'll talk about then on the carbon side, photosynthesis through solar-induced fluorescence, and about hyperspectral measurements, which can get you a variety of plant functional traits. So I have all of one slide on remote sensing of evapotranspiration. I promised you high resolution at some of these measurement types, and you see that in this map right here. This is from the eco-stress sensor, which is on board the International Space Station right now. This is LA in a pretty sort of hot day last year. You can obviously get these same measurements also in more remote areas where there's perhaps a bit more trees, but there's actually plenty of trees in LA as well. And this hopefully just gives you a quick visual sense of 30 meters relative to sort of what you'll see is basically tens of kilometers that we get with microwaves. So on the one hand, ET estimates from space are super exciting because they're really high resolution, so they really might be helpful with things like the scaling challenge that we've talked about. On the other hand, they're really indirect, right? So most of these estimates are based primarily on estimating land surface temperature. And then to get from land surface temperature to the actual flux of water, there's no sort of magical remote sensing trick. What you do is these estimates just link to a variety of different sort of hydrological, basically sort of biogeophysical models. And there's a wide variety of data sets out there that use a wide variety of different estimates. Just like the microwaves do, all of these have pros and cons. But that's just something to be aware of. So that's the flux of water. In terms of the sort of flux of carbon at the land surface, there's been a lot of really exciting developments over the last 10 years or so in remote sensing of photosynthesis through increasing development of these solar-induced or sun-induced fluorescence measurements. So I'm not really going to go through this, right? But the basic idea, most of you are probably familiar with this, is that in the photosynthesis process, we end up with excess excited photons. Plants can partially get rid of those through non-photochemical quenching, or they can emit these fluorescence photons, which are now able to be sensed, even if it's somewhat noisily, from space. And so you get these maps that look something like this, where this is now a machine learning-based estimate of photosynthesis, and then we look at SIF, and we see to first order there's a pretty good match. Even better than that, but I didn't want to put it on the slide because there's so many of them. There's lots and lots of careful studies where people have compared SIF measurements to flux towers and really seen a very good match. So this is exciting. And on top of the initial excitement over the last 10 years, there's even more excitement because we've now, over the last couple of years, really gotten sort of arguably a fairly big game changer in SIF measurements through the Tropomi mission that's been up for, this is late 2017, I think, the launch, so about 2 years now. We really increased the resolution from sort of roughly 50 kilometers of the old SIF data sets to now 100 times more data, closer to 0.05 degrees or about 5 kilometers, depending on where you are. So last but not least on this little blitz tour of remote sensing technologies, I wanted to briefly mention hyperspectral measurements. which can give you a variety of things. So this give you so many different variables. I've just summarized it as plant traits. Right, and just to sort of understand the basic idea behind hyperspectral measurements, right, Ashley mentioned multispectral before. Many of you might be familiar with things like MODIS or Landsat, right? So you get, maybe 10-ish different measurements at different electromagnetic wavelengths, different corresponding to those wavelengths frequency bands, right? For reference, microwaves usually only have one wavelength. Sometimes you get 2 or 3 on a sensor if you're lucky. Hyperspectral sensors, very excitingly, put together hundreds of these spectrometers. So the exciting thing about hyperspectral sensors is they put together basically hundreds of spectrometers at different wavelengths to allow you to not just have 5, 8, 10 measurements, but really pick up full spectra of a given area on the land surface. And the exciting thing is different components of the plants have totally different ratios of reflectances between different wavelengths. And so when you have enough of these different measurements, you can actually back out the composition of the landscape along a variety of different axes, as you see on the right-hand side of the slide here. And these hyperspectral measurements are, again, also really nice because they can get you high resolution. As you see this, I forgot to put a scale bar on here, but this figure in the lower left is at Jasper Ridge, out near Stanford. Is about, I think in this particular case, about a 500 by 500 meter focus area, where you can really see across the fairly diverse landscape of Jasper Ridge how things like the carbon ratio, the nitrogen ratio of the leaves, and actually the water content vary. So that's our little blitzkrieg, but that timer is winding down. Maybe not quite a krieg, as Natan is laughing in the audience there, but that timer is winding down. So let's just focus on microwaves here. And I just want to do, again, a tiny bit of housekeeping for everybody, because there's multiple different microwave sensors. You basically, on the one hand, have radiometers, which look at passive gray body emission, right? The same emission that my shirt and everything around us is giving off. And those are measured. And then on the radar side, we have an active measurement of an electromagnetic wave that's sent out, and we look at how much is scattered back as captured by this so-called backscattering coefficient. And so you'll see me go back and forth, and there's opportunities and advantages, disadvantages to both in the rest of this talk. One of the key differences between them is on the side of the resolution. So radiometers are generally a little bit coarser resolution than the scatterometers in space. Depends a lot on the individual sensor, but we're generally looking at 10 kilometers is really an aggressively high resolution, right? Maybe 25 to 50 seems more reasonable. A lot of scatterometers are maybe a little bit finer but similar, but the nice thing about radar, of which I should clarify a scatterometer as an example, is you can also use synthetic aperture techniques, basically taking advantage of the Doppler effect to artificially increase your resolution a lot. So with synthetic aperture radar sensors, you can get to resolutions of maybe a little bit more than 100 meters or so, depending on sensor and how much noise you're willing to accept. So there's possibilities there, but for now, we have these coarser measurements than you might be used to. But in my opinion, still super exciting measurements because, again, they are sensitive to water content across much of the canopy, even in cloudy conditions. Right? So let's just talk a little bit about what exactly these sensors see. And the good news is it's actually very similar between radars and radiometers. There's basically 3 different main ways, main terms that can contribute to your observations. On the one hand, you can have things like this schematic arrow on the right, where a radar wave is scattered directly by the vegetation at perhaps different heights. You can say, okay, well, not all of the electromagnetic wave is scattered back by the vegetation. Some of it passes through and is attenuated, but still that electromagnetic wave also hits the soil, or maybe there's a gap between the canopy and things like that. And then you also get scattering at the soil interface. That scattering depends on soil moisture, which is why a lot of these sensors, at least on the land side, have been developed for the sake of measuring soil moisture more accurately. And then you get these interaction terms as well. And the nice thing is, every single one of these three terms is attenuated and affected by the vegetation in a manner that depends on the water content. And that's true for radar, as you see in this schematic. But you can imagine for radiometry, it's a very similar concept. Just basically subtract one of the errors for simplicity to first order. OK, so great. We can go and delve into the literature of what these electromagnetic scattering models and microwave frequencies look like a little bit more. And you usually get something like this, where the real idea is to say, OK, we're going to represent vegetation as a collection of cylinders and leaves. And the exact amount of scattering or the exact amount of emission depends basically on the geometry, the sort of electromagnetic geometry of these different trees, right? Do you have thick stems? Do you have tall stems? What is the angular distribution of the leaves relative to the stem? And so you get these models with parameter tables that look something like this, which I don't know if in the back you can even read those words on there, but the whole point is that they're very unwieldy and there's a lot of parameters. And it seems at first blush like it might not be relevant at all to sensing water content. The good news for us is that aside from being sensitive to this geometry, you're also sensitive to the dielectric constant of each of those components, essentially sort of the capacitance, or the potential for capacitance in them. And that dielectric constant is much higher for water than it is for most other components of soil and vegetation. And that's what gets us our sensitivity. In fact, if you look at one of the very earliest models, right, in 1978, Atema and Ulabi for radar backscattering of vegetation, it actually uses this phrase as a water cloud. It literally just says we have a collection of droplets, and that's how we're going to represent the vegetation. And then we put some multiplicative parameter in front of things, depending on what exactly the vegetation geometry is. And that sort of practice is still in use today and has held up over generations of technology development since. And you see that. So on the radiometry side, this attenuation factor, this transmittivity gamma, is considered to be within an exponent linearly proportional to the vegetation water content, that mass of water per unit area as encapsulated by number of droplets in the vegetation with some linear scaling parameter. Radar models, you have a similar thing. Maybe there's an exponent in there that's not always exactly equal to 1, but often will hover around there. And so we can nicely collapse all of that geometry effect to some of these scaling parameters, but that doesn't mean that those scaling parameters are well understood. I just want to make a quick note here while I sort of have equations for radiometry and for radar up, that there's, again, lots and lots of advantages and different advantages to radars and radiometry. But for some of you who have been keeping an eye on this literature to different extents, you might see that at least in the, shall we say, ecosystem use literature, there's a lot of emphasis in the last couple of years of VOD, a lot of very exciting things happening in the radar world as well, especially by Sasan and others. But there seems to be sort of the sense that VOD is more accessible. You got to be a little bit careful with that because that basically is partially just a byproduct of how easy the data sets are to handle. So you got these long equations up here that I'm not really going to go through, and the details are not really important. But these are sort of canonical equations for our brightness temperatures that a radiometer might measure, and for the backscattering coefficient that a radar might measure. And you see each of them depends on that same gamma coefficient, that transmissivity coefficient that I mentioned in the last slide. That's a function of water content. you're faced with these observations, and then to figure out what the vegetation signal is and how that distinguishes from the soil moisture signal, you have to isolate that gamma from your raw measurements. There's different tricks for this, but I've just highlighted in green how many of the variables in these equations are sensitive to vegetation geometry. And for the radiometry, that nicely collapses to one parameter. For radar, it collapses to, in this case, 4. It's actually more than that because there's different polarizations as well, which is this PQ here. And so VOD data sets are much easier to retrieve, arguably so. That does not mean that those retrievals are any less noisy or any more correct, right? In this case, a scattering albedo, that B parameter, those things are also unknown. So just be careful on that. But one effect of this is that you'll see we have many more results for radiometry on the plant hydraulic side than for radar. Okay, so far I've mostly talked about basically electromagnetic theory on why we should be sensitive to vegetation water content with our microwave measurements. Is that actually something that holds up in reality? Well, one way to do that is to go look at these field studies where instead of immediately going all the way up to space, you put a radiometer or a radar on, in this case, a truck. You can put them on towers, some sort of field scale study. And because of these microwave applications for soil moisture, actually people have been doing those studies for many, many decades now, and we have a whole variety of them. So this is an example for soybeans, right? We see just here one study where we see the very nice linear relationship between destructive gravimetric measurements of water content of the soybeans and the optical depth that you retrieve from the radiometer. You can do a similar experiment for radar. Again, a little bit more noise, but fairly nice, at least at lower water content levels, very nice one-to-one relationship between what we predict based on inverting our radar measurements and what's actually measured gravimetrically in the field. These are just two examples for soybeans. There's, like I said, many, many studies like this done for soil moisture applications. Here's just one list of them. Actually, that's now 30 years out of date. We can add a whole bunch more to this, but even back then, there was a long list. And for those of you close enough to the front who can read this, right, here are the land cover types associated with each of those field experiments. They're almost all in agricultural areas. We have far fewer in forests. And one of the reasons for that is that it's simply much harder to do these destructive measurements to actually figure out how much water is in different components of a tree or a tree in its entirety. So the couple that are out there sort of hidden in the literature, a lot of them are also, as you'll see later on, just water of different components of the tree rather than necessarily everything that you could be seeing. But still, we got some theory. And we got a bunch of measurements in other land cover types, so we can be optimistic about all of this applying to forests as well. So what I'll assume for the rest of this talk is basically that we can trust the relationship between the microwave measurements and the actual water content, right, that VWC. But I just want to kind of keep re-emphasizing this point about structure. Is not completely lost, right? So this is a meta-analysis of different studies that have looked at that geometric factor relating the relationship between the optical depth that we see with the radiometers, or also definable by the radars, and the water content. And it's a little bit misleading, or you have to be a little bit careful in reading this plot because this is a semi-log scale, right? But if we pick a given frequency that a given sensor might be working at, there's still a lot of variability depending on exactly what measurement, and presumably at least largely depending on the geometry of the particular vegetation that you're looking at that relationship. Still, we got water content across most of the ecosystem. That's really hard to get otherwise. Okay, and then just to push this a little bit further into how we can think about it in the context of looking at water stress responses, even beyond the geometry influence, we can think of the water content in a tree as being influenced by lots of different things, and as something that you can interpret multiple different ways. The easiest way that I have found to think about it is through this simple equation. Right? If we think about the mass of water per area, we can decompose that as the relative mass of water compared to the total mass of water in the vegetation times the total mass in the vegetation, right? This is just algebra. But then we can really see this as a relative water content times what is the total above ground biomass of that vegetation. So you start to see that aside from geometry, biomass variations also influence what we're seeing, which can be a blessing or a curse depending on what you're interested in. If you're interested in mortality, maybe there's actually some opportunity there and not just some annoying thing you have to disentangle. But in the meantime, for the next couple slides, I will treat it as some annoying thing that you have to disentangle. Because the question then is, beyond water content, right, can we go to variables that are most relevant to plant hydraulic studies? So I think just about every single one of the other talks this morning mentioned these pressure volume curves, right? Relationships between the water potential, in this case in the xylem, and the actual relative fraction of water in those components. Those differ by species. This is just one example. You've seen other slides before. And I think as Pierre nicely emphasized, a lot of those are somewhat nonlinear, right? And so there's this question, okay, we have water content. We would really like to get to water potential as being dramatically more useful for modeling and studying efforts. How important is this nonlinearity? How much does it get in the way? There's issues of scaling that we've talked about. How much does all of that stuff get in the way? Well, we can just look at it, right? So here's one example of a study that did that. This was my former postdoc Mustafa Momin et al. Together with Kim and others, we went and just compared VOD data directly from space to all the places where we could get our hands on leaf water potential measurements at 3 different sites in this particular paper. One of them I've just shown here. This is Sevieta in New Mexico, as Pierre also showed data from. And you see that over the course of 3 summers here, you get sort of a fairly nice correspondence between the leaf water that we upscale across the 2 main species at this site and what we measure at the VOD. Now this is a data set that's gridded to 25 kilometer resolution, but the underlying native resolution is coarser. We have, I don't remember the exact number, but maybe 10 leaves per species here. There's a lot of noise that you would expect in here. And so to me, the fact that we see any sort of relationship at all, R squared of 0.82 here, if we also account for the small influence of LAI, is a real testament to the potential of these data sets despite all the uncertainties. We can look at other harder places than just Sevieta with 2 species. Again, this is Spaceborne data, really coarse, related to a couple trees measured in situ, but at Manaus, at a much more diverse location than New Mexico. So these are just 3 relatively dominant species at this site. This is work by Tim Van Emmerik and Susan Steele-Dunn. And you see there's not that much data, but when we actually compare Spaceborne measurements of backscatter in this site in the tropics, where very nicely, because we're in the wet tropics, there's so much vegetation that the electromagnetic waves are no longer sensitive to soil moisture, and we don't have to worry about that. So we can just say, use this as a direct proxy for that vegetation contribution, that vegetation water content that we're supposedly seeing with the radars. And then we relate it to, or the authors here related that to dendrometer measurements on a couple of these different species. So some sense of changes in stem water storage. And you see, there's some nonlinearity here. These are just a couple points. But again, given the amount of variability involved, the fact that you have any relationship at all, I think is pretty exciting. So those are sort of seasonal variations, right? Pierre nicely introduced the idea of seasonal as well as diurnal variations. So we can ask ourselves, do we see diurnal variations, or can we learn something from diurnal variations as well? And here we can sort of start to play tricks with the orbits that the spaceborne sensors usually have. So most spaceborne sensors are in what's known as a sun-synchronous orbit, right? And they're doing things like what you can see here in the red line is just sort of the path that a different sensor is tracing out on Earth underneath it. And a lot of these sun-synchronous, or these sun-synchronous orbits rather, not a lot of them, what they're doing is they're revolving around the Earth in such a path that they are rotating at the same time as the Earth is rotating around itself in such a way that they're nicely following along with the time zones. So each orbit takes about 90 minutes or so. And as you do that, you're slightly changing your longitude such that by the time you get back to the same latitude as you started out with in this example, you've now passed 90 minutes later, but you've gotten to a different longitude such that you still have the same local time, right? So that makes interpreting these data about a gazillion times easier, right? Because now you can say that basically everywhere except for the highest, most latitudes, for a given sensor, we're always sensing, say, 6 a.m. no matter where we are. Now these traces have to go both north and south. So usually you also have, well not usually, if the data is downloaded from the sensor, you also have a measurement 12 hours apart. So for a given sensor, the 6 a.m. is very common, very nicely for us. Lots of these sensors, you have measurements at 6 a.m. on the one side everywhere, and 6 p.m. as well. Again, nicely for us, given the importance, I think, as Patrick mentioned, of midday measurements. We also have 1:30 a.m. and p.m. as very useful. And so we can start to look at that. I'm just going to race through this because I'm running out of time. But these are just 2 sample pixels, and you see this in lots of places around the world, that to first order, the diurnal dynamics make sense, aka to first order, things are wetter at night. The dashed line here, when you see things at 1:30 a.m., then you see at 1:30 p.m. in midday during the depression. Actually figuring out how that diurnal dynamics varies across time obviously depends a lot of the details of the plant hydraulics. But you see in this example pixel in India, for example, to first order that difference decreases a little bit as things get wetter and you have faster conductance. And we can go a step further than that through the magic of the orbit on the International Space Station, which is not in a sun-synchronous low Earth orbit. It is in such an orbit that basically the local time of day of each measurement changes. Now, if you focus on a specific point, then it takes sort of a while across different times when the sensor comes back to see that point, to really trace out all of the different times of day that are possible. But if you average over enough measurements, you can really see a complete diurnal cycle. And so that's what's shown in this slide here. On the left, we have an average across the rainforest in Central Africa. Again, dense vegetation. Don't have to worry about soil moisture. And so we can use the RapidScato radar that has this orbit. And you see diurnal dynamics that kind of make sense, right? Pierre talked about refilling at night. You see basically right up until pre-dawn, you're increasing in backscatter. And then as soon as the sun comes out, you drop. You can argue a little bit about how sensitive this sort of middle, relatively flat point is to your exact averaging. But maybe there's just a model siesta in there, right? And then at night after sunset, you can start going back up. Probably pushing it a little bit, but the basic dynamics are there. We can do that at Manaus as well. This is only a single pixel. This is only 1 month, so it's a lot noisier. But again, sort of promising, hopeful things that still have details to be worked out. We have, in collaboration with the folks from SMAP, we had a field campaign out this summer, so finally moving away from some of these scaling issues from when we look at things from space. But where we actually had, at Harvard Forest, a radiometer looking out over about 20 by 20 meters of relatively homogeneous red oaks. And then we did 3 days of 90-minute interval pressure chamber measurements. Those are pretty long days. But we got some data, we got some nice psychrometer data, and just as a first order, if you look at the average diurnal cycle, it matches up pretty well. There's a little bit of a difference here that we're still trying to understand. Some of that might be just due to vertical height that we're sampling at, and sort of differences across the entire canopy in terms of how fast things drop and what the actual integrated average VOD height is that we're seeing. I'm going to run out of time, but I just want to, for those of you in the audience who have seen this, want to briefly mention that there's a lot of studies out there that use VOD as an indicator of biomass alone. So some of you might wonder what is going on, with also thinking that it's water content. And so, just sampled, ignore the offers to protect the innocent, but shown some titles here. And just want to understand a little bit how that fits into our picture here, right? What's happening is these studies generally use maps like this, like Sasan's map of static biomass across the world, and then relate that to average VOD. So they're looking at how biomass influences these measurements in space. And when you see that, I don't want to sort of not emphasize this enough, there is a strong biomass influence on what we sense with VOD. This is a really strong relationship. But to go from there to actually interpreting all VOD dynamics as representing biomass dynamics requires you to trade space for time. And if you think back to our equation from before, it basically requires you assume that this relative water content term is constant. That might sort of average out in the wash when you look at an average in time across the entire globe. But I think I've got a pretty friendly audience here that says, well, maybe that's not always true. So with that, just want to put some of these conclusions up, right? We have biomass as a confounding factor. We have scale. But really, the key point is we have lots and lots of partial indirect avenues of evidence from different angles to suggest that there's information content here, but actually understanding the limits of that information content, understanding the effect of vegetation, geometry, scaling, things like that. There's still a lot of open questions where hopefully you guys come in. I'm going to leave this up during the questions because I'm out of time in case someone wants to ask me about other specific issues that could influence these measurements. And the biggest that I think we'll have to talk about more is this issue of how different microwave frequencies penetrate the canopy to different extents in different kinds of ecosystems. But thanks a lot.